Okay, in part 2, we'll look at how enzymes are functioning as catalysts. Let's look at some general properties of enzymes. First of all, they catalyze the making and breaking of covalent bonds inside the cell. Then they increase the rate of reaction by 100,000 to 100 billion times faster. And thirdly, they allow reaction to occur under milder reaction conditions, such as near to neutral pH and low temperatures, such as our body temperature or even in room temperature. And fourth, enzymes have very high specificity. For instance, they can distinguish in between different types of optical isomers, such as the D isomer or the L isomer. And last but not least, enzymes regulate the biochemical reactions and pathways inside the cell. Okay, before we discuss any further about enzymatic reaction, we'll look at what is a chemical reaction. So in any chemical reaction, you have reactants that are turning into product molecules. So in a reversible reaction like this, the products can also be converted back into reactants. But over a long period, the reaction will reach in a state known as equilibrium, by which there is no net change in the concentration of products and reactants. So at equilibrium, the ratio of the product divided by reactants is known as the KEQ, or the equilibrium constant. And in this case, the KEQ is equal to 98 times 98 divided by 2 times 2, which is about 2400. So KEQ is a constant, and this value is dependent on the energy values of both reactants and products. To be more specific, the equilibrium constant of a reaction KEQ is correlated with the energy released by that reaction. So that is called the change in Gibbs free energy, or delta G, which can be calculated by using this formula. Delta G is equal to negative R times T times ln KEQ, by which R is gas constant, while T is temperature measured in Kelvin. So in our previous example, KEQ is 2400. So fitting in this value into the formula, you will get delta G as negative 19.3 kilojoule per mole. Whenever the value is negative, it means that this reaction will happen spontaneously. Okay, then you might be asking, why do we need enzymes in biology? If delta G is negative, it means that reaction can happen spontaneously or naturally without the help or without booster from any other components. However, it doesn't say how fast that reaction is. In fact, reactions without enzymes can be very slow. It might take hours, it might take days, weeks, or even months to complete. Why is that so? That is because the intermediate transition state in chemical reactions has a much higher energy level than the reactants. So energy is required to generate this transition state before substrates can be converted into product molecule. And this energy is known as the activation energy. So it is the energy supplied to the substrates in order to reach the transition state. And activation energy will determine the reaction rate. So when the substrates are converted into products, the free energy release is called delta G, which will determine the equilibrium constant, or the KEQ. But when the enzyme is present, transition state has a lower energy level because the substrates are now bound inside the active site. So that increases the reaction rate and also reducing the activation energy. And as a matter of fact, all enzymes or all catalysts will reduce the activation energy. Now you have learned that in biochemical reaction, something is needed to reduce the activation energy. In chemistry, we call it catalysts, but in biochemistry, our catalysts are enzymes, which are mainly composed of proteins, while some they are composed of DNAs or RNA. So by binding the reactant, enzyme changes the environment and hence reducing the activation energy. Thus, you can now convert glucose into carbon dioxide and water quietly without flame, but just using enzymes. Because now you have a much lower activation energy, in which your body temperature at 37 degrees Celsius is sufficient to kickstart the reaction. But how does enzyme work? In order for enzyme to function, they need to form a complex with the substrate molecule before they can turn substrate into product molecules. And the formation of this ES complex leads to the transition state. And there are three important points about enzymes. Number one, enzymes increase the speed by which equilibrium is arrived, but they do not change the amount of products and substrates at equilibrium. So in other words, enzymes do not change the equilibrium constant of a reaction. And last but not least, enzyme is unaltered on the completion of reaction, and therefore they can be reused or recycled. Now you might be asking, what does an ES complex do? First, ES complex holds substrate out of the aqueous solution. So by doing this, it's actually concentrating the substrate within the active site. Second, it holds substrate in specific orientation, which is closer to the transition state to allow reaction to occur. Remember, without the transition state, no substrate will be turned into product molecules. And third, it reduces ability of free rotation and molecular collisions with the non-reactive atoms. In other words, this helps to reduce unwanted reactions. And last but not least, it allows an altered local environment within the active site, such as a different ionic strength, an altered pH, or it can add or remove hydrogen bond to the substrates. In a nutshell, enzymes facilitate the formation of transition state. So what is transition state actually? Well, you can say that in transition state, the reactants are no longer substrates, but they have yet to become product molecules. And when you have more transition state, you're going to get more product formation simply because that the conversion of transition state into product molecule is an irreversible process. So the transition state sounds pretty much like a romantic relationship where you and your friends are more than friends but less than a couple. Okay, very briefly now, we will look at the catalysis mechanisms by enzymes. First of all, proximity. So in order for an enzymatic reaction to occur, the substrate molecules must come close together. Then orientation. So you'll need the correct orientation for the chemical reaction to happen. And during the binding, they actually strain or distort the chemical bonds so that the old bonds are broken while new chemical bonds can be formed. So there are actually two general types of enzyme catalysis, either an acid-base catalysis or a covalent catalysis. So in the acid-base catalysis, one amino acid will serve as a weak acid that donates proton, while another amino acid residue will act as a weak base that will generate hydroxyls. While in the covalent catalysis, a temporary covalent bond in between enzyme or substrate will be formed. So in this process, it involves the donation or acceptance of electrons, while in the acid-base catalysis, you have the donation or the acceptance of either protons or hydroxyl groups. And next, we will look at the specificity of enzymes. Okay, before a reaction can happen, both enzyme and substrate must interact in close proximity and they must interact in the right orientation. And now you also learn that enzymes show specificity by only binding to certain substrates. And this specificity is due to the interactions in between specific structural regions on the substrates and on the enzyme as well. And the specific structure on the enzyme is known as active site. In other words, we can say that both enzyme and substrates are showing complementary structures. And the specificity of the enzyme is dependent on the specific shape of the active site and some other factors like chemical forces such as hydrogen bonds and so on. The specificity of enzyme can be explained in two ways. The first one was proposed by Fischer in 1894, which is the lock and key model. So in this model, the binding site has an exact complementary shape to the substrate. Well, a second theory was proposed by Koshland in 1958, which is the induced fit model. So in this model, the active site is slightly flexible because the contact between part of the binding site and the substrate can induce a change in shape of the active site to fit the substrate. Okay, let's look at the first model, which is the lock and key model. So in this analogy, enzyme is the lock while substrate is the key. So only the right key can enter into the right lock, and therefore only the right substrate can enter into the enzyme. And when this happens, it forms an enzyme substrate complex, which is the transition state. And in this stage, you might have acid-base catalysis or the covalent catalysis involving the donation or acceptance of electrons to form new chemical bonds. And subsequently, substrates are converted into final products and the enzyme can be reused again. While in the second model, you have the induced fit theory, so in which the shape of the active site is not completely complementary to the shape of substrate. But what happens is that when they come close together, the binding of substrate induces a change of shape of the active site. This is what we call the induced fit process. And then again, you will get this ES complex in the transition state, and substrate will be converted into final products. Okay, enzyme specificity can happen at many levels. The first type is absolute specificity, which means one and only one specific substrate, such as urea and urease. So urease will not act on other substrate except urea. While the second level is bond specific. So in this level, enzyme will identify and act on a specific bond type, such as the carbon, oxygen, and carbon bond in an ester molecule. So R1 and R2 can be of any length. They can be any random hydrocarbon molecules, but it doesn't matter because this enzyme esterase will always act on an ester molecule as long as they can detect this carbon, oxygen, and carbon bond setting. Then you also have the group-specific enzymes such as hexokinase. So whenever you see the term kinase, it is always associated with phosphate group. So here, a hexokinase will transfer phosphate group to a hexose, which is a 6-carbon sugar molecule. But as you can see, both sugars are having different configurations as the position of the hydroxyl group are different. But again, it doesn't matter because for this enzyme, it is specific only towards the phosphate group that it transfers, and the hexose can be of any isomer. While the last level is stereospecific, in which the enzyme can distinguish on the stereoisomers such as the dextroisomer or the levoisomers. Okay, brain teaser for today. One slide in this lecture is wrong, and it is wrong in its concept, which is formula related. Please try to identify where is the mistake.